8. Heredity. We have seen that reproductive processes give rise to new individuals that are similar but stubbornly different. We have discussed how to some amount of variation is produced even during asexual reproduction, and the number of successful variations are maximized by the process of sexual reproduction. If we observe a field of sugar cane, we find very little variation among the individual plants. But in a number of animals, including human beings, which reproduce sexually, quite distinct variation are visible among different individuals. In this chapter, we shall be studying the mechanism by which variation are created and inherited. 8.1 Accumulation of variation during reproduction. Inheritance from the previous generation provides both a common basic body design and subtle changes in it for the next generation. Now think about what would happen when this new generation in its turn reproduces. The second generation will have differences what they inherit from the first generation, as well as newly created differences. Figure 8.1. Figure 8.1 Creation of diversity over succeeding generation. The original organism at the top will give rise to, say, two individuals similar in body design but with stable differences. Each of them in turn will give rise to two individuals in the next generations. Each of the four individuals in the bottom row will be different from each other. While some of these differences will be unique, others will be inherited from their respective parents, who were different from each other. Figure 8.1 would represent a situation if a single individual reproduces, as happens in asexual reproduction. If one bacterium divides and then the resultant two bacteria divide again, the four individual bacteria generated would be very similar. There would be only very minor differences between them, generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated, as we will see when we discuss the rules of inheritance. Do all these variations in a species have equal chances of surviving in the environment in which they find themselves? Obviously not. Depending on the nature of variation, different individuals would have different kinds of advantages. Bacteria that can withstand heat will survive better in a heat wave. As we have discussed earlier, selection of variants by environmental factors form the basis of evolutionary processes, as we will discuss in later section. 8.2 Heredity. The most obvious outcome of the reproduction process will remain the generation of individuals of similar design. The rules of heredity determine the process by which traits and characters are reliably inherited. Let us take a closer look at these rules. Inherited traits. What exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human beings. However, if does not look exactly like its parents or human populations show a very deal of variation. Activity 8.1. Observe the ear of all the students in the class. Prepare a list of students having free or attached ear lobes and calculate the percentage of students having each. Figure 8.2. Figure 8.2. Free and attached earlobes. The lower part of the ear, called the earlobe, is closely attached to the side of the head in some of us and not in others. Free and attached earlobes are two variants found in human population. Find out about the earlobes of the parents of each student in the class. Correlate the earlobe type of each student with that of their parents. Based on this evidence suggests a possible rule for the inheritance of earlobe type. Rules of inheritance of traits, Mendel's contributions. The rules of inheritance of each traits in human beings are related to the fact that both the father and the mother contribute particularly equal amounts of genetic material to the child. This means that each trait can be influenced by both parental and maternal DNA. Thus, for each trait, there will be two variables of each child. What will then the trait seen in the child be? Mendel, Mendel, George John Mendel. Mendel was educated in a monastery and went on to study science and mathematics at the University of Vienna. Failure of examination for a teaching certificate did not suppress his zeal for scientific quest. He went back to his monastery and started growing peas. Many others had studied the inheritance of traits in pea, the other organism, earlier, but Mendel blended his knowledge of science and mathematics. And what is the first one to keep count of individual exhibited a particular trait in each generation? This helped him to arrive at the law of inheritance. Mendel worked out the main rule of such inheritance, and it is interesting to look at some of his experiments from more than a century ago. Mendel used a number of contrasting visible characters of garden pea: round, wrinkled seeds, tall or short plants, white or violet flowers, and so on. He looked pea plants with different characteristics. A tall plant and a short plant produced progamy by crossing them and calculated the percentage of tall or short progeny. In the first place, there were no halfway characteristics in this first generation of or F1 progeny. No medium height plants. All plants were tall. This meant that only one of the parental traits was seen, not some mixture of the two. So the next question was, were the tall plants in the F1 generation exactly as the same as the tall plants of the parent generation? Mendelian experiment test this by getting both the parental plants and these F1 tall plants to reproduce by self-pollination. The progeny of the parental plants are, of course, all tall. However, the second generation or F2 progeny of the F1 tall plants are not all tall. Instead, one quarter of them are short. This indicates that both of the tallness and shortness traits were inherited in the F1 plants, but only the trait was expressed. This led Mendel to propose that two copies of factor, now called genes, controlling traits are present in sexually reproducing organisms. These two may be identical or may be different depending on the percentage. A pattern of inheritance can be worked out with the assumption as shown as figure 8.3. Figure 8.3 Inheritance of traits over two generations. Activity 8.2. In figure 8.3, what experiment would we do to confirm that the F2 generation did in fact have a 1 ratio 2 ratio 1 ratio of capital T, capital T and small t trait combinations? In this explanation, both capital T and capital T are tall plants, while only small t is a short plant. In other words, a single copy of capital T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits like capital T are called dominant traits, while those that behave like small t are called recessive traits. Work out which trait would be considered dominant and which one recessive in figure 8.4. What happens when pea plants showing two different characteristics rather than just one are breed with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what happens when these F1 progeny are used to generate F2 progeny by self-pollination? A Mendelian experiment will find what some F2 progeny are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new combinations. Some of them would be tall but have wrinkled seeds, while other would be short, but have brown seeds. You can see as to how new combination of traits are formed in F2 offsprings when factors controlling for seed shape and seed color recombine to form zygote leading to form F2 offspring. Figure 8.5. Figure 8.5 independent inheritance of two separate traits, shape and color of seeds. The ratio of this 9 ratio 3 ratio 3 ratio 1. Thus, the all short or tall traits and the round seeds or wrinkle seeds traits are independently inherited. How do these traits get expressed? How does the mechanism of heredity work? Cellular DNA is the information source for making proteins in the cell. A section of DNA that provides information for one protein is called the gene for that protein. How do proteins control the characteristics that we are discussing here? Let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. Plant height can thus depend on the amount of a particular plant hormone. The amount of the plant hormone made will depend on the efficiency of the process for making it. Consider now an enzyme that is important for this process. If this enzyme works efficiently, a lot of hormone will be made and the plant will be tall. If the gene for the enzyme has an alteration that makes the enzymes less efficient, the amount of hormones will be less and the plant will be short. Thus, gene control characteristics or traits. If the inheritations of Mendelian experiments we have been discussing are correct, then both parents must be contributing equally to the DNA of the progeny during sexual reproduction. We have discussed this issue in the previous chapter. If both parents can be helped determine the trait in the progeny, both parents must be contributing a copy of the same gene. This means that each pea plant must have two sets of all genes, one inherited from each parent. For this mechanism to work, each germ cell must have only one gene set. How do germ cells make a single set of genes from the normal two copies that all other cells in the body have? If progeny plants inherited a single whole gene set from each parent, then the experiment explained in figure 8.5 cannot work. This is because the characteristics capital R and small y would then be linked to each other and cannot be independently inherited. This is explained by the fact that each gene set is present not as a single long thread of DNA but as separate independent pieces, each called a chromosome. Thus, each cell will have two copies of each chromosome, one each from the male and female parents. Every germ cell will take one chromosome from each pair of these, may be either maternal or paternal origin. When two germ cells combine, they will restore the normal number of chromosomes in the progeny, ensuring the stability of DNA of the species. Such a mechanism of inheritance explains the result of the Mendelian experiments and is used by all sexually reproducing organisms. But asexually reproducing organisms also follow similar rule of inheritance. Can we work out how their inheritance might work? Sex determination. We have discussed the idea that the two sexes participating in sexual reproduction must be somewhat different from each other for a number of reasons. How is the sex of a newborn individual determined? Different species use very different strategies for this. Some rely entirely on environmental cues. Thus, in some animals, like a few reptiles, the temperature and which fertilized egg are kept determine whether the animals developing in the eggs will be male or female. In other animals, such as snails, individuals can change sex, indicated the sex is not genetically determined. However, in human beings, the sex of the individual is largely genetically determined. In the other word, the genes inherited from our parents decide whether we will be boys or girls. But so far, we have assumed that similar gene sets are inherited from both parents. If is the case, how can genetic inheritance determine sex? The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and paternal copy, and we have 22 such pair. But one pair, called the sex chromosome, is odd in not always being a perfect pair. Women have a perfect pair of sex chromosomes, both called X, but men have a mismatched pair, if with some if, in which one is a normal sized X while the other is a short one called Y. So, women are XX while men are XY. Now, can we work out what the inheritance pattern of X and Y will be? As figure 8.6 shows, half of children will be boys and half will be girls. All children of inherit an X chromosome from their mother, regardless of whether they are boys or girls. Thus, the sex of children will be determined by what they inherit from their father. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy. Figure 8.6, sex determination in human beings.